And our goal is to discover novel small molecules that bind to targets. And initially, we worked with a lot of epigenetic targets. So we had a lot of lysine methyltransferases, arginine methyltransferases, and binding proteins. But now we are moving into the E3 ligases, and that's a very exciting field. So we take a protein family approach, which means that we are interested in, for example, proteins that methylate K36, lysine 36 on the histone free, and there are several of them, for example, NSD1, 2, and 3. And we want to express the protein, screen it against the library, and identify hits, and then test with hits in cells. And the chemical probe is defined by a potent, selective, and cell-active compound. So we basically check those boxes, and then our goal is to release it into the academic community so we can do the biology, so we can use it to discover what these proteins do. And one of the success stories that is in this symposium is the NSD2 probe that binds to a domain in the protein, which is a small domain, and the Structural Genomics Consortium spent quite a few years studying that little domain. It's a PWWP domain that is required for NSD2 when binding to its product, which is a dimethylated K36, and propagate the mark or do the processes that it's required to do. So once we have this compound, then we can disrupt the interaction. But what we can also do is design a degrader from that compound. And that's an ideal situation to be, because we're not inhibiting the catalytic function. We're basically using it as a handle to recruit. So Promega technologies are great, and then as we joke in the lab, Promega saved the whole program of measuring binding interactions. So measuring protein-protein interactions is difficult. And then when I was doing it in the lab, it was immunoprecipitation techniques or something that was cumbersome, and it really wasn't stoichiometric, and it wasn't kinetic. It had a lot of problems. Now, the live cell measuring target engagement, the way you guys do it with NanoBRET, we adopted the technology right from the start because it worked, and I wish we had it earlier. I especially like the NanoBRET because there are other configurations where you have split nanoluck, you have different kinds. But here, because we're working with the compounds, and the compounds, well, as one of the chemists like, and I always quote this, every compound will have a nightlife that you may never know about. It's gonna go and do something in cells, and you just come across and, oh, it kills cells. Well, what does it mean? So doing a ratiometric approach where you have the luciferase signal and then you have your fluorescent signal, so the blue and red, is great. And we adopted that for quite a few targets. We even went into these difficult assays that we have right now of measuring the ubiquitin binding domain to binding to ubiquitin. And we talked to your scientists so many times that they helped us solve many problems. And then the tracer technology. We don't do bromodomains where the tracers are absolutely awesome, or kinases. So we're just getting into some of our targets where we could use your tracer technology, but it would be nice to have more versatile, more of those tracers, because not every protein family will have a generic bromosporine or so, tools like that. Technical expertise, ideas, and the reagents, right? So just having a really robust assay is very, very helpful, because then we have to do less troubleshooting ourselves.